Okay, putting a wrap on Chapter 2, Cognitive Neuroscience. As promised in the initial part, we will talk about methods for understanding what's going on in the brain while people are engaged in cognitive tasks. And so I have a little list here. As we mentioned in the previous video, Hubel and Weisel's discovery of the simple cells in the cat's visual cortex is an example of what's called single cell recording. So, as I described it, we're going to insert a very small microelectrode into the cell of a neuron. And whenever it fires an action potential, this electrode is a sensor essentially, and it's going to detect that electrical activity of that one individual cell. So we can then determine under what conditions is this cell firing an action potential very, very quickly. That's how we know when a cell is stimulated, by the way. It's not just one occasional action potential, but when it's receiving consistent excitatory input, it's going to fire its action potentials at a very, very rapid rate. Neurons can generally fire up to 1,000 times per second. So we're going to see this rapid spike in the rate of firing. We might also see inhibition. So what's important here is to realize that when a neuron is not being excited, it's not quiet all the time. You can have random, spontaneous action potentials. And so that's what we're going to see is that when a neuron's just at rest and there's no particular stimulation, you will have the occasional spontaneous spikes coming out of these neurons. When there's excitation, you see an increase in that firing rate relative to the background spontaneous firing rate. When there's inhibition, you see the exact opposite. You see a decrease in the neural firing rate relative to the background rate or baseline rate. So that's useful, right? So that we can determine under what conditions. I think we got people working on my ceiling above me here, so sorry about the banging. Uh, so under you can know under what conditions a cell is being excited versus inhibited by detecting these immediate changes in that individual cell's firing rate. Now, on the other hand, we can check neural firing rates not for individual cells, but for whole populations, whole groups of neurons. And this is where EEG is useful. So you may have seen EEG caps before, right? These sensors that are now just embedded into a cap that you wear on your scalp. And these are detecting the same kind of electrical activity that the single cell recording techniques are, but they're not able to isolate it to one single neuron, right? What they're doing is they're detecting when groups of neurons all fire action potentials in synchrony, that generates an electrical field that's strong enough to expand out from the skull, and these sensors in the EEG cap can detect those electrical fields. And so this is now getting us these, like I said, whole populations of neurons are active all at the same time. When this occurs in response to a stimulus, it's called an ERP or an event-related potential. And the idea we're referring to the potential here as an action potential, but it's event-related, meaning it's triggered by some particular stimulus. Of course, we can also do other things like look at just brainwave activities. We can talk about alpha and beta rhythms and stuff like that, brainwaves that vary in their frequency and synchrony, and things of that might change as a result of different kinds of cognitive tasks as well. fMRI is our next one up here. So the F is pretty important here. MRI by itself is an example of what's called a structural imaging. And so if someone has like torn ligaments in their knee, we can do structural MRI to identify which ligaments are torn and this maybe even the severity of that. If someone has something going on with their brain, we can do a structural MRI of their brain and we can identify brain tumors or developmental venous anomalies or anything like that, right? Structural defects in the brain. But functional imaging is different. Functional imaging looks at changes over time. And in this case, what's happening is that we're going to look at changes in blood flow over time. This is called the BOLD response for the blood oxygen level response, right? So what's happening is that different regions of the brain become active during a task. Neurons are going to fire faster action potentials. And after a neuron fires an action potential, it gets into that refractory period and it requires the sodium potassium pump to reset the neuron back to its resting state, right? And this is a very metabolically demanding process. So when cells become active and their sodium potassium pumps become more active, they require more energy, and that comes from blood flow. And blood contains, especially the idea of oxygen here, blood contains red blood cells that have iron in them, right? But they have the oxygen bound to them as well. And so the idea is that this oxygenated blood is going to be flowing to different regions of the brain. And then it gets used and it becomes deoxygenated blood. And we can see this change from oxygenated to deoxygenated blood. And we know that certain neurons in certain regions of the brain are becoming a bit more active because there's more of that kind of conversion taking place. And it changes the magnetic output of um, the blood, right? So that's what's happening, right? The MRI is a great big magnet, so it's going to detect the paramagnetism of the iron atoms in the oxygenated blood, and we can figure out which areas of the brain become more active during a cognitive task. So if we give someone a task that is designed to isolate, say, memory functions or perceptual functions or language functions or anything like that, we can look to see which regions of the brain become active during that task, and that's going to tell us maybe that those regions of the brain are a bit more important and a bit more relevant for that particular kind of task. FNIRS is a very similar kind of thing here, but instead of using a big magnet for an MRI, we're going to use a cap that might look a bit more like an EEG cap, but instead of detecting electrical potentials, this detects infrared light or near infrared light, technically speaking here. So these caps actually have two different kinds of instruments embedded in them. One is a small near infrared light emitting diode that shoots near infrared light through the cranium and then into the brain. And then what happens is it's going to hit oxygenated blood that reflects this light because it's the right color, right? Red blood. And so again, it reflects that light back out. And now there are light sensitive cells that detect this reflected light. So wherever we have oxygenated blood flowing in the brain, it's going to reflect more of this light, and we're going to be able to detect that using the cap here. Now, this only works at a relatively surface level, right? This light doesn't, you can't shoot these light rays way down deep into the brain and detect activity of subcortical structures the way you can with fMRI. So there is a little bit of a disadvantage here to FNIRS, which is that you can only get the outer cortex activity here. Why would we do this then if it's not as good as fMRI? Well, it's a whole lot cheaper for starters, right? To buy an MRI machine is going to be millions of dollars, or you can rent time in a hospital, but then you're only going to get in there at limited times of the day when they don't need it for other patients, which might be like 3 or 4 in the morning, and you still cost a lot of money to rent per hour. Whereas you can buy a pretty good FNIRS system for your neuroscience lab for just a couple $1,000s, and then it's yours to use as you want. It's also much more portable. In MRI, people have to be very still, and you can't have anything metal in the magnetic field. That's dangerous, right? So with FNIRS, people can be a little bit more mobile. You don't want them moving around a lot, but they can move around and they can sit upright in a chair and do things at a computer. You don't have to worry about metal or anything like that. Another technique is through simulation techniques. So transcranial magnetic stimulation, that's TMS, or transcranial DC current stimulation, TDCS. What's happening here is that we're either going to use a magnetic field or we're just going to use direct current, and then we're going to stimulate the brain through these electrical currents, right? So a magnetic field essentially is just going to cause action potentials to happen in different neurons, right? And that's also what the DC signal will do as well. And so we can then directly stimulate neurons. But this is a very non-invasive procedure that uses an external emitter that uses these fields to induce the action potentials in the neurons like this, right? So it's very, like I said, non-invasive. And so then the idea is that once we do this, uh, we can see what happens. Like what happens if I force these neurons to become active in a person, right? And does this trigger certain memories or certain perceptual experiences or anything like that? Or we can also, by the way, we can suppress their activity as well. And so if I suppress a certain neural region in the brain, and then I ask someone to try to do a task and they can't do it because that region is suppressed, that teaches us something about the importance of that part of the brain for that task, right? Here, I just have some pictures. So we'll go through these relatively quickly. This is showing fMRI. Here on the top, we have activity during hand movement. So a person is moving their hand. We have on the top moving their right hand in a healthy subject, healthy meaning no brain damage here. We see the left motor and left sensory areas primarily activated right here, right? On the ipsilateral, we see some activation. So this would be the right side of the brain. You remember that um, control for the brain is generally contralateral, right? So the left side of the brain controls the right hand. So we see that during hand movement, a person is going to have motor area activation that indicates that this is controlling the hand, but you're also going to feel that. So you see this sensory areas in the parietal lobe also active here. On the ipsilateral side, there's a little activation, but it's much reduced, of course. Why is there any ipsilateral activation? And notice it's only in the motor region. Um, that's because our hands are used to working together. So whenever the right hand is doing something, the left hand's motor control circuitry is going to be somewhat active because, again, we're used to doing things with both hands a lot. So there needs to be some coordination, even when the two hands are not both working simultaneously. Now, here we have a person with a stroke, right? And so what happens in the stroke patient is that the parietal activation is now much reduced and it's less active. And the ipsilateral activation is also very much active here. So we see less coordination between the two sides of the brain now. This is going to tell us some things too about then what happens during stroke. At the bottom, we have a blind participant who is using their fingers to read Braille. And so not only do we see some of the motor and somatosensory areas active, but curiously, we're also seeing some occipital lobe activation, and that's the visual areas of the brain, as if maybe there's some visualization that can occur here. This is FNIRS. So you see here the cap that I was talking about, and you see that there's two different kinds of technologies embedded into the cap. We have the light emitting diodes, and then we have the light sensitive detectors. So you can see that diagram at the top. You have a detector and a source. So the source, beams the near infrared light in there, and the detectors detect the reflected light. And we can get similar kinds of maps of showing activation areas during different kinds of tasks. One last thing to say about cognitive neuroscience is this field called neuropsychology. So what might happen is that let's say we do structural imaging and we find a lesion. That is, we find that a person has had a stroke, or maybe they've had some sort of brain trauma, what we might call a closed head injury from maybe a really serious concussion. And we might find some damage to a region of the brain, but it's a very small, subtle kind of thing. Well, one of the things that we can do is we want to understand, well, now that we know that this person has this small amount of damage to this part of the brain, what kind of impairment are they going to have? And if we can figure that out, then obviously that's going to tell us something about what that region of the brain does. Okay, because if the impairment is something that's specific to reaching and controlling the hands, or maybe it's something specific to reading or language, then of course, obviously, we can map that out by mapping the location of that lesion to a specific cognitive impairment. But the way this works is in neuropsychology is something like this. What we need a battery of tests, right? So once we say that we got a person who has a lesion in their brain and we want to know what's impacted here, well, we might need to give them a very large battery of different kinds of neuropsychological tests. We can give them here an IQ test that measures different kinds of functioning, verbal functioning, perceptual, working memory, and processing speed. We can give a memory scale. We've got a verbal learning test. We've got the complex figure test that tests for visual spatial ability. The trail making test also tests for spatial ability, but also search and scanning, and so on and so forth, right? All these different kinds of cognitive tests measure different aspects of cognition. And so we can see which one of these are impacted when we observe that lesion. But here's something that's even maybe a little bit more critical. Sometimes when people have a brain injury, the lesion is not visible in imaging. Sometimes it is very, very small and very, very subtle, and we can do MRI, structural MRI, and we can do CT scans, and we just simply don't see anything. And so we think, well, maybe there's nothing wrong with them. Well, wait a minute, let's find out. Let's go back, and even though we don't know where the lesion is, we're going to administer this battery of neuropsych tests, and we're going to see which of these tests shows some form of impairment. And then once we figure out, oh, okay, now I know where what the impairment is, that's going to give me some clues about where in the brain I can look in for maybe a little more carefully to see what's going on, and maybe there's a lesion in that particular region that we didn't notice before, right? So it helps guide your uh, imaging done later. Keep in mind, this is not nearly a complete list of neuropsych tests. This is a pretty small list, but it kind of gives you a good overview, broad overview of all of the different kinds of cognitive tasks that could be tested here. And that now is it for our chapter 2. So on to the next chapter in the next week.